Hello, everyone, and welcome to today's webinar, Optimizing High-Content Screening Tools for Physiologically Relevant 3D in Vitro Models. I am Michelle Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Molecular Devices. To learn more, please visit www.moleculardevices.com. Let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speakers, Dan O'Connor, North America BioImaging Director, Molecular Devices, Elodie Vandenhaute, Study Director at HCS Pharma, and Zied Souguir, Senior Scientist, HCS Pharma. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Our speakers will now begin their presentations.
Thanks, Michelle. Hi, this is Dan O'Connor. I'm the North America Director for Bioimaging with Molecular Devices. Today I'll be talking about overcoming challenges in automated high-throughput 3D imaging using high-content imaging. As you can see from this slide, there's a variety of 3D applications ranging from zebrafish, neurite outgrowth, organoids, C. elegans, Drosophila, 3D whole slide imaging, and spheroids, to name a few. We'll be going through each of these applications and how we can overcome challenges with solutions of imaging. Here's a typical 3D cell culture steps for creating bioimaging assays in 3D. As you can see on the left, you culture spheroids, treat with compounds, stain for markers, acquire a 3D cell image, and then analyze with your high-content imaging software. Today, we'll be talking strictly about these last four, acquiring 3D cell images and analyzing. Here are some challenges that you will encounter with high-throughput 3D imaging. I have listed 5 of the top challenges that I believe are creating issues with large 3D imaging assays. 3D imaging creates undesirable, out of focus, or hazy images that can skew image analysis. 6 samples have difficulties with light penetrating through the entire tissue. Reliable automated focusing on 3D plate types, such as organ on the chip and U-bottom. Plate acquisition time is long when you have to do multiple fields of view and Z-stacking. Data sets are large and complex. Large data sets can take hours to analyze, becoming a bottleneck. Exporting data to a 3D imaging software can be tedious, cumbersome, and inconsistent. Standalone 3D imaging software is not designed for multi-well, multi-plate data visualization and high-content data mining. The first challenge, 3D imaging creates undesirable, out of focus, or hazy images that can skew image analysis. You can see on the left, we have a wide field image and a confocal image. The confocal image cleans up that haze or out of focus part of the image very well. On the right, you see the analysis. And in confocal, we count over 30% more of the nuclei, making it a much more accurate quantification with confocal. We have 2 solutions with molecular devices to tackle this. If you have a wide field system, you have a on-the-fly 2D deconvolution that allows haze cleanup. You can do this 2D deconvolution or digital confocal in both wide field and confocal. We also have a hardware solution that is a spinning disk confocal technology that is optimized for high resolution, tissue penetrance, and high throughput. We have recently launched high performance customizations on the IXM confocal that now includes high intensity lasers, auto dispensing water immersion objectives, and a proprietary confocal disk module for decreased pinhole crosstalk and increased tissue penetrance. As you can see in this slide, we lay out the optical path of the IXM confocal with these new high-performance customizations. It is now optimized for automated high-throughput 3D imaging assays with our water immersion objectives, 20, 40, and 60x, that reduces light refraction for brighter intensity at lower exposure times. We also have the deep tissue penetrating confocal disk module that allows greater depth into tissue and reduces crosstalk to improve autofocus light suppression. This confocal disk module has a low light transmittance, but we make up with high intensity lasers for 5 or 8 channels. These lasers are 1,000 milliwatt per channel and offer best-in-class light excitation. The second challenge for 3D image acquisition is light penetrance into thick 3D samples. We just discussed the confocal disk module that increases light penetrance into thick tissues with a specialized disk geometry designed for reduced pinhole crosstalk and autofocus light suppression. In this image, you can see with a 60 micron pinhole confocal spinning disk versus the deep tissue confocal disk module. Another challenge is reliable automated focusing on 3D plate types, such as organ-on-chip and U-bottom. Our solution is to have a robust high-speed laser and image autofocus. This allows you to consistently and reliably focus on each round bottom plate in 96 wells or 384 wells. On the right, you see our laser autofocus and our software autofocus mechanisms. The key feature here is that the system finds the plate bottom, sets that plate bottom thickness, and then finds the well bottom and sets that offset. This allows for a very robust laser autofocus and focus mechanism. Third challenge is plate acquisition time is long compared to 2D. Typically in a well plate, you will have your objects of interest scattered throughout the well. The only way to capture each of these objects is to scan the entire well, as you can see with this grid overlay. Our solution is to do a quick ID targeted imaging. The system will automatically find XY positions of objects of interest with a low magnification, automatically switch to a high magnification, such as 40x in this case, and go to those points of interest, and the objects are always in the center of the region of interest. This allows for faster acquisition, less image storage, and less images to analyze. The last challenge here is data sets are large and complex. Again, large data sets can take hours to analyze, becoming a bottleneck, and exporting data to a 3D imaging software can be tedious. A standalone 3D imaging software is not designed for multi-well, multi-plate data visualization. Our solution is to have an integrated 3D analysis with single interface, reducing time to discovery. We can now quantify 3D structures with volume, intensity, and distance measurements, all with one interface end-to-end software. Here's a couple of snapshots with our 3D image analysis. These algorithms identify cells in organoids and detect volumes. These images were captured with a 10x plan Apo 0.45 numerical aperture objective in 60 micron pinhole confocal with Z-stack of 23 Z-planes at 10 micron steps. These objects of interest were about 250 microns. You can see in the software can automatically identify each cell at each different Z-plane. This allows the software to not count the cell more than once. We can also detect volumes in the bottom right picture. Here's a 3D viewer to visualize XYZ colocalization and organoid volumes. I will play a quick video that shows the features of the 3D viewer software. We also lay out another challenge of data sets are large and complex. It takes hours to become a bottleneck. Our solution is to accelerate analysis speeds by up to 40x with multi-threaded parallel processing. With our MetExpress PowerCore software, we can now go from hours to minutes eliminating 3D analysis as your bottleneck. You can also run multiple image express systems in parallel. As you can see, we utilize the Intel processors for multi-threaded parallel processing. To continue the challenge of data sets are large and complex, we also tackle this by having an integrated solution for bioinformatics called Acuity Express. This allows for 3D phenotypic data mining and visualization. We can do plots, self-organizing maps, principal component analysis, curve fitting, all automatically with the touch of a button. We have additional resources such as ebooks and our new 3D web page that I encourage you to click and explore. Our 3D ebooks are a great resource for all the information that we covered in this webinar. Next up, we have HCS Pharma, which is a wonderful partner of Molecular Devices. They are a new technology, 3D application technology driver that has CRO services that can maximize the rentability of your studies by enabling more parameter extractions in less time. They will go through their technology in more detail. Thank you.
Thank you, Dan, for the introduction. So I'm Elodie. I'm working in HCS Pharma company, and I'm here today with Zied Souguir, our expert in biomaterials, who is also the inventor of biomimetic technology that we will detail later on. I'm working as a study director, and I will talk to you about HCS Pharma and the biological results linked to biomimetic technology. So in this second part, we will tell you how to create physiologically relevant 3D in vitro models for the screening of molecules using biomimesis. So HCS Pharma is a company localized in France, in Lille, more precisely. We are equipped with two cell culture labs, one automation and cell imaging lab that you can see on this picture, and one production chemical lab for the creation of biomimesis plates. We are also equipped with robots in standalone, and we will talk to you more detail about the two image express microconfocal systems that we have integrated on the platform. We provide services in safety and efficacy assays in medium and high throughput. We also are providing services linked to cell assay development, proof of concept and mechanism of actions, as well as consulting in pharmacology, cosmetology, and nutraceutics fields. And as now you know, we are selling also products, biomimesis plates, that we also are using in-house for 3D cell culture studies. On this slide, so you can see different pictures of the platform that was designed and integrated by molecular devices, so different views. You can see the two image express microconfocal systems that we have. One on the left being thermostated for lifestyle imaging. One robotic arm taking the plates from one device to another. One pipettic system on the left for 96 and 384 well plates from CBO, 3 different incubators, 1 plate sealer, and also 1 plate washer. So everything is integrated on this platform that we are using for studies, screening of compounds, for example, for the pharmaceutical industry. So we just want to introduce you a little bit about the high failure rate in drug discovery, because as you know, 90%, you may know, 90% of drugs fail in very late stage, meaning in clinical trials, in particular in phase 2 clinical trials. And this is due to lack of efficacy. And we think that this lack of efficacy in the clinical trials is due to a lack of relevant in vitro models, human in vitro models in the preclinical stages. In fact, most of the screenings in the early stages of drug discovery are performed on 2D cell cultures, meaning so cells grown on plastic as monolayers most of the time. The point is that they differentiate within a few days, while in reality the cells are grown in a complex environment. Different cell types are together. They are all embedded in extracellular matrix, and they are also blood vessels, which is obviously not the case in 2D. That's why we think that we should go further in 3D cell culture models, and that's why we are using biomimesis for in vitro studies. So our point of view is that we should reproduce 3D cell culture with at least many cells and with the ECM, meaning the extracellular matrix, with the appropriate physical chemical properties, and that's why we are using biomimesis. Now I will let Zied talking about this technology in more detail.
Thank you, Elodie. To introduce biomimesis technology and the added value of our technology, we have to present the existing technology and with their advantages and limitations. The first class are the scaffold-free models based on the formation of monospheroid using external stress by the modification of the form and surface treatment for Euler plate or gravity for hanging drop and using metallic nanoparticles for magnetization. The advantages of the technology are their ease to use and their adaptability for screening. But the lack of specific microenvironment is a major limit and disadvantage in this system. For the classification of the second class based on a matrix as a support for cell culture, we will focus on the nature and the physicochemical properties of different models. We find the classic system based on hydrogel, for example, thermosensitive hydrogel as a nitrogel, and other hydrogel based on biopolymer or synthetic polymer. Among the advantages of this system are their cost and the adaptability for screening, but their need of preparation before use and the bad diffusion of growth factor and or oxygen due to the encapsulation of cells added the model are oncogenic or non-physiologic microenvironment are the major limitation of the system. The second kind are solid scaffold based on synthetic plastic like polymer as polystyrene and other polymer. The systems present a cost and ready-to-use advantage. The limitation of this system are the absence of specific cell matrix interaction and the difficulty of their use in imaging system. The third class is the hydroscaffold model, and for us, hydroscaffold are 3D system combining hydrogel and solid scaffold behaviors, and we consider deserialized organ or tissues as hydroscaffold. This system presents a real physiologic microenvironment, but their high cost, their poor reproducibility, and the difficulty of their use in screening and imaging system are the main limitation. To overcome all the limitation of this last model, we propose the solution with our technology Biomimetic hydroscaffold, and the technology ready to use was cost-effective, usable and screening, and with a good reproducibility and versatile system. And Biomimetic, it was developed to better mimic the HCS. But what we need to reproduce HCM today, all scientists agree that the important role of the HCM to maintain the structure of tissue and to define the cell microenvironment, and HCM is a key determinant of cell behavior and function. And so HCM composition is tissue specific. Indeed, for each organ or tissue, we find the main compounds with different amount. The HCM are composed generally by glycans, aminoglycans like uric acid, and structural proteins or adhesion proteins like collagen or fibronectin or laminin. The role and uric acid to HA has multiple functions in HCM to maintain the structure and as a reservoir for water and growth factor and receptors. The second important and specific characteristic of each HCM is the mechanical properties or the elastic modulus that define the stiffness or elasticity of HCM. For healthy or pathologic organ, and the composition and the rigidity of the HCM are also indicator of the organ pathologic or healthy organ. And to reproduce the HCM, we need to know the composition and the mechanical properties. And now I'm going to explain how we develop biomimesis. Biomimesis is carried out by cross-linking reaction of uric acid with collagen 1, 4, and 6 using ADH as a cross-linker to obtain at first an hydrogel, but the hydrogel is not suitable for cell culture due to the fast degradation. And the innovation in HCS Pharma, we have developed a new process to improve the mechanical property of biomimesis and to transform hydrogel to hydroscaffold. And we see in the picture the same composition on comparison in vivo system with a deserialized organ. And just to explain why biomimesis is hydroscaffold, when you added medium to biomimesis, the hydroscaffold swell in presence in medium, and when we zoom, we observe under microscopy the presence of fiber and translucent due to presence of hydrogel. And then we have the two behavior of like solid scaffold and hydrogel. And now biomimesis is hydroscaffold. Biomimesis is marketed in precast matrix in 96 well plate with different format plate and demand, and we can modify the thickness of the matrix for hydroscaffold for application imaging or another. And today is the ready to use only technology based on ionic acid and SAM compounds proposed in the market. We have developed four projects at first per organ. We have biomimesis brain, biomimesis liver, and biomimesis adipose tissue. And per pathology, we have biomimesis oncology used for different stage of cancer. And now I let Elodie present some proof of concept and biological result with biomimesis technology.
Thank you, Zied. So I will take over, and first with biomimesis liver. This matrix is composed of hyaluronic acid grafted with HCS adenosine peptide, galactosamine, collagen 1, and collagen 4, with an elastic modulus of 0.6 kilopascal. This matrix has already been validated for the culture of cell lines like HG2 cell line and HG cell line. It has also been used for the culture of primary human hepatocytes. And last but not least, this matrix has also been used for the differentiation of induced pluripotent stem cells into mature hepatocytes. I will show you a couple of results. First, with biomimesis liver and primary human hepatocytes. As you can see on the electron microscopy pictures, the matrix by itself is close in its structure to the decellularized liver extracellular matrix. You can see the hyaluronic sheets and the interlaced fibers of collagen. On the right, you can observe primary human hepatocytes that have the form of spheroids within the matrix. We have characterized the cells in these conditions and compared it to 2D culture in collagen 1 sandwich condition. The interesting thing is that albumin secretion is much higher when the cells are cultivated in biomimesis liver. And this higher production of albumin is maintained over time. We have tested until 28 days and 1 month. And again, the CYP activities have been quantified, and CYP3A4, for example, is its activity is doubled in biomimesis compared to 2D. CYP1A1 and 1A2 activity is multiplied by 3. And CYP2B6, you have more than 8-fold increase between 2D and 3D for the basal activity of the CYP. So this shows that hepatocytes grown in biomimesis liver maintained in functionality for, at least 1 month, and suggest that they are close to the in vivo situation, at least closer than 2D culture. HG2 cell line has been cultivated in biomimesis for the study of the cholestatic effect of compounds. Chlorpromazine is a compound that can induce a destabilization of biliary canaliculi, which is known as the cholestatic effect. You have the X staining of the spheroids together with the green staining of CD-FDA, which is a substrate for this biliary canaliculi. And you can see with the increased concentration of chlorpromazine that you have a destabilization in the structure of the canaliculi, And this could even be quantified using MetaExpress granularity application module. So you can see the graph on the right that is quantifying the effect, the procholestatic effect of chlorpromazine. Moreover, ApoRG cell line has been cultivated in Biomimesis liver to check for the pro-steatotic effect. The steatosis is the increased accumulation of lipids. And again, this is a parameter that you can easily analyze with image analysis using adipored staining, for example. We have tested different compounds that you can see on this table that are known to induce steatosis or not. And we compared 2D condition with spheroids obtained with ultra-low attachment plates and with biomimesis culture in 3D, and compared, in fact, the responses and the steatotic effect. As you can see on the table, the 2D, all the compounds have shown pro-steatotic effects with at least a double, most of the time, a double increase in the accumulation of lipids. With ULA plates, you still have this high increase in the accumulation of lipids, but even if there is a little shift in the concentrations. And with biomimesis, you have, we observed less impact of the compounds on the accumulation of lipids, on the steatosis effect. Again, when we could see this effect, it was with higher concentrations, much higher concentrations, and even for the two last compounds, so tretinoin and methotrexate, no effect could be observed. So this seems that there is a lower steatosis effect on the cells in biomimesis liver. The cell survival was better in these conditions. So what we could conclude is that in 2D, it can give warnings according to the toxicity or safety of molecules, but it's not predictive of what's happening in human beings, because all these drugs are taken by humans because they are approved medicines. So biomimesis seems more predictive in the human effect. I will talk to you about the differentiation of iPSCs into hepatocytes in biomimesis liver. This is a collaboration with Institut du Thorax in Nantes. This is a PhD student who has done this work. We have compared the differentiation in 2D versus the differentiation in biomimesis in 3D, and I've done an extensive RNA-seq study to check the expression of genes. You have all the examples here of genes that have been upregulated in 3D, and they are dealing with bile metabolism, fatty acid metabolism, cholesterol, lipoprotein, glucose, insulin, cytochrome, and xenobiotic related metabolism, which is of great interest. It shows that the cells are mature. And we have characterized the spheroids that we have obtained in this condition, and this organoid contains obviously hepatocytes. So you can see on the picture thanks to the albumin staining in green. But very interestingly, we also know that there are some other cells, like stellate cells, as you can see with the red staining, and also endothelial cells. This was also suggested by the RNA-seq study because we knew that some genes were not from hepatocytes. So we have a complete organoid obtained from this differentiation. And we are now characterizing the spheroids in terms of CYP activity, for example, and metabolism. I just give an example here with CYP3A4 and CYP2C9 that we see are active in the spheroids. All these results suggest that the differentiation of iPSCs into hepatocytes is great in these conditions, and so studies are ongoing in this field. I will now move to biomimesis adipose tissue. Biomimesis adipose tissue is composed of hyaluronic acid and RGDS adhesion peptide, and collagen 1 and 6. Its elastic modulus is of 0.45 kilopascal. And this matrix has already been validated for the culture of primary human adipocytes of cell lines that can be differentiated into adipocytes. And our collaborators have also used it for the differentiation of iPSCs into mature human adipocytes. I will show you a couple of results, but if you need more information, just be aware that the publication was done by Louis in 2017 in Biotechnology and Bioengineering Journal. Again, the structure of biomimesis adipose tissue is really close to the in vivo decellularized human adipose tissue extracellular matrix, as you can see on these pictures. Interestingly, the cells are organized as spheroids, as grapes, here at day 14, as seen by electron microscopy, which looks like the structure that you can also observe in vivo. And on the right, the images show you the spheroids within biomimesis, and the staining by adipored, but shown here in green, shows an accumulation, normal accumulation of lipids, of triglycerides more particularly, within the spheroids of adipocytes, which is what we can expect from such cells. We also compared this condition with 2D conditions. And again, you can see on this graph that there is a higher accumulation of the triglycerides in the cells within biomimesis. Again, this differentiation is maintained for at least 1 month. In 2D, you can see from the pictures that it's really difficult to maintain a complete culture expressing and showing good accumulation of lipids, unlike in 3D on the right in biomimesis. Also, some genes have been validated as differentiation markers, but I won't show them here. You might have more information in the publication if needed. Just for your information, biomimesis adipose tissue has also been successfully tested for the culture of sebocytes, in particular human iPSC-derived sebocytes. This is a collaboration that we have with Phenocell. Thanks to this picture, you can see that the sebocytes also accumulate lipids, as seen in green. And on the right, they also express MUC1 as a sign of good differentiation within biomimesis. So this shows that the cells can be also maintained at least for 1 month, since these pictures were taken at day 28 after seeding. So I'm done with biomimesis adipose tissue, so I will move to biomimesis oncology. So the commercial matrix is composed of hyaluronic acid and collagen 1, with an elastic modulus of 0.8 kilopascal. And biomimesis oncology has been validated for the culture of cancer cell lines in general, I mean adherent cells, and also for the culture of cancer cells from patients. I will show you some results about drug testing in biomimesis oncology. Here you can HT29 cells with live dead green and dead cells are red. We tested different concentration of the anti-cancer drug 5-fluorouracil, 5-FU. And thanks to imaging, we could design the EC50 curves, and we can see from the graph that there is a shift in these EC50 curves between 2D and 3D in biomimesis, suggesting again that the responses are closer to in vivo situation in terms of responses to drugs. So this type of study is done on other cell types, and as other screenings are ongoing. And now I will present you the results regarding how we can play around with the matrix and with its composition and physical properties, because it's something that can be done with biomimesis but not necessarily with other types of systems. Here we played with the elastic modulus of biomimesis and checked the culture and the growth of HCT 116 cells within few days. You can see the X staining of the spheroids in the matrix. And interestingly, we could observe that the increased elastic modulus with increased collagen 1 is linked to higher cell proliferation in biomimesis. So this also can be linked to what has already been described in other studies and also in vivo regarding the impact of the physical properties of the matrix on the cell proliferation and the impact of mechanical stress on the cells. We have played around again with the composition and stiffness of the matrix, but this time by putting laminin into the matrix. Here is a study performed on the breast cancer cells MDA-MG 231. We've stained UX and the calcium green, meaning the living and proliferating cells are in green. And we have checked the evolution of the area of the spheroids, meaning the growth of the cells, a long time, for 1 month. And here you have again an impact of the elastic modulus, but importantly, in the presence of laminin, we were decreasing, in fact, the cell growth, contrary to what we have observed before. Just the presence of laminin had an impact, a negative impact on the growth of the spheroids over time. So this is quite interesting, and again, which is in line with what has already been seen in other studies. And also interestingly, the expression of CD44 was checked again a long time. CD44 here, which is seen in red, and the composition and stiffness of the matrix seems not to have an impact on the expression of CD44. Just in all conditions, it seems to have increased a long time. So the matrix stiffness and the presence of laminin had no impact on the expression of this marker. I would like to finish with biomimesis oncology by telling you that it can also be used in vivo thanks to its properties, because it is solid. It can be easily grafted in mice, and it was compared to classical injection of cancer cells in mice, and the grafting was much higher, was much successful with biomimesis, with 100% success compared to 63% success in just with the injection of cancer cells. So it can also be used in vivo with good engraftment of the cells within the surrounding tissues. The last matrix I will present is biomimesis brain. This one is composed of hyaluronic acid, collagen 4, RGDS adhesion peptide, and a cationic biopolymer. The elastic modulus is quite low. It's 0.1 kilopascal. To be in line with the in vivo, the fact that the brain is really smooth. This matrix has been validated for the culture of different cells like lumens, immortalized human dopaminergic neuronal precursor cells, which can be differentiated into mature dopaminergic neurons, but also for SY5Y cells in spheroids, and also for the culture of human primary astrocytes. But this is one of our last matrices, so we are still characterizing this. So other validations will come shortly, for example, with primary human neurons. So different cell types like lumen cells can be grown in biomimesis for, again, 1 month, as you can see on this picture with UX and beta-3 tubulin staining. Obviously, lumen cells cannot be cultivated for so long in 2D. And primary human astrocytes, as you can see with the green staining in green staining, sorry. You can maintain again primary human astrocytes for 1 month. we checked again the impact of drugs on Lume's cells, this time in biomimesis brain, and the impact of MPP+ and erastine that are known as Parkinson's disease's inducers. In particular, erastine is responsible for a specific type of cell death, ferroptosis. And we compared 2D and 3D culture in biomimesis. We tested them at different concentrations to grow the curves, so the impact on cell death. And there is a little shift in the curve with MPP+, suggesting that the higher concentration should be achieved to induce cell death in biomimesis. And more importantly, regarding erastine, we had difficulty to see the high cytotoxic effect of erastine at the tested concentrations, because in 2D, less than 1 micromolar is necessary to kill all the cells, and we could not observe any cytotoxic effect at the tested concentration within biomimesis. So it seems that we have a lower sensitivity of the cells in biomimesis, and we are going on with investigations to know why and what's the mechanism behind. I will finish with biomimesis types of plates because now we have biomimesis in 384 well plates, which is very convenient for high throughput screening studies. We have great reproducibility between plates and also within one plate regarding the 3D cell cultures achieved in these conditions. So screening is currently ongoing with biomimesis oncology to check the effect of anti-cancer drugs and to maybe find new drugs in libraries. So this means that biomimesis is available for light screenings of molecules. I will finish just with a few words on 3D cell culture flow, because obviously, since we are using cells in 3D, we need to make the proper picture acquisition, which is possible with our MetaExpress microconfocal systems. And then we need to reconstruct the spheroids that we get, or the structures that we can observe in 3D, before extracting interesting parameters and interpreting the biological results. And this is what can be achieved with the 3D analysis module of MetaExpress. So in the software, there is a 3D analysis custom module. And for example, a find spherical objects algorithm is very efficient. So I will send you the video. So as you can see on this video, you can easily, in fact, image the spheroids. This spheroid was not obtained in biomimesis because it is a unique spheroid. It was in ultra-low attachment plates. You can see the nuclei of the cells, and you can check the numbers. You can check the size of the spheroids. You can also do analysis of volume. Thanks to this module. And I will finish on what to remember about biomimesis technology. So biomimesis technology is the only ready-to-use hydroscaffold composed of hyaluronic acid and other ECM components on the market. It is natural and physiological. because hyaluronic acid is under its physiological form, and you are in presence of undenaturated collagen. This matrix is scalable and versatile. You can have it in different formats. It is compatible with most, if not all, common downstream analysis techniques, even if I haven't given more details, but just know that you can do PCRs, you can take the cells for flow cytometry after culture, etc. Primary cells maintain their phenotype longer in biomimesis, thus allowing long-term experiments. I mean, chronic treatments are allowed. And biomimesis can also be used in vivo as well. Don't hesitate to check our website for more information, or to send any other question to our email hello@biomimesis.com. So I would like to thank you for your attention, and before going to your questions, we would like to thank Guillaume Frugier and Cédric Point for Molecular Devices for our collaboration with them, and to Sarah Piper for organizing this webinar. Thank you.
Thank you.
Thank you, Elodie, Zied, and Dan, for your informative presentations. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. Okay, first question is, let's see. Okay, it looks like we may have answered this in the presentation, but just to recap, have you already developed a Biomimesis 384 well plate?
Yes, so Biomimesis has been developed also in this format. So if you need such format, you can order this. Yeah.
Great, thank you. Great. Okay, next question. What do you mean by it can be applied? Sorry, one moment. What do you mean by it can be applied to in vivo?
We just mean that it can be used for grafting in mice. I mean, you can seed cells within the matrices and just take the matrix, it's solid. You can put it and graft it in mice, and this has already been done with a collaborator. So this is the way I wanted to say when I said you can use it in vivo. So for example, cancer cell grafting. I hope it's clear, but yeah.
Great, thank you. All right, next question. How have you done your fixation and permeabilization before your IF?
So it depends on the protocol, but most of the time we are using most of the time formalin for fixation, and for the permeabilization we use either twin or Triton X100. So you can use both depending on your staining and even I guess others because the matrix is quite stable in such classical protocols, I would say. So
thank you, Elodie. All right, next question. How many cell images can catch from 384 well for imaging analysis?
Michelle.
Yeah.
Oh, sorry. Yeah. Go ahead, Elodie. That's fine.
No. It's your question. No problem.
Yeah. Okay. I was curious because it was the question I was not exactly sure on what is being asked, but how many cell images can catch per 3D 4-well plate?
Correct.
If we do, we can have several different objectives. If we have a low magnification objective, for example, a 4x objective on our systems, we can capture the entire well. So you can take 384 images per well, or you can go all the way up to 100x magnification, which would then be about 100 images per well, and in that case, that'd be about 38,000 images per 384 well plate. I hope I answered that correctly. I was a little confused on the exact intent of the answer or question.
Great, thank you, Dan. Okay, next question. Let's see, wow, we're getting so many great questions. Okay, what is the cost for a 96 well plate?
So there is a difference between the different matrices in terms of composition, but it's around between 200, around 300 euros per plate. But obviously, since we can also do customized matrices, we can also do specific quotations for very specific matrices. But it's around 250, 300 euros per plate.
Great, thank you. Okay, next question. Is there any development of biomimicry for muscle cells?
Sorry?
We have some collaborators try smooth muscle cells, but at the moment we haven't a result.
Thank you. All right, next question. Let's see. Okay, next question. How do you make fixation and permeabilization of 3D spheroids?
It was the same fixation and permeabilization I've just talked about. Formalin for the fixation, triton or twin for permeabilization for most of the images. Yeah.
Thank you. Okay, the imaging of 3D spheroids showed nuclei only around the outside of the sphere. Have you used any techniques to more fully capture the inside of the spheroids, such as clearing?
I think I can answer this. So yes, we've definitely used techniques for tissue clearing. In fact, it's a great way to see deeper into tissue. There's several different tissue clearing methods out there. We have worked with Visicol in the past, but yes, this is a great way for seeing clearly into thicker 3D organoids.
Great, thank you. How do you manage the imaging system?
I can answer that as well. I believe this is in reference to maybe IQ-OQ or GXP environment. Our imagers don't have an actual GXP or IQ-OQ. However, we have several customers that we can customize and lock down certain features for auditing purposes and CFR Part 11 compliancy.
Great, thank you. All right, it looks like we have time for one more question. Looks like this one is for Elodie and Zied. Have you ever worked with smooth muscle cells in your matrices?
I think we have answered already to this question, if I'm not wrong. We haven't here in the lab worked with smooth muscle cells, but some collaborators have done so. We cannot give you the results or how it worked, but I mean, there's no reason why smooth muscle cells will not grow in biomimesis, but we haven't tried here.
Great, thank you. And it looks like we have a request for publications, and we will follow up with your question after the webinar is finished. So thank you for that one.
Yeah.
So thank you again, Elodie, Zied, and Dan, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Molecular Devices, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their questions. Any questions we did not have time for today and those submitted during the on-demand period will be addressed by the speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.